Hello everyone, my name is Daiyun Huang. I'm a third-year PhD student from Xi'an Jiaotong Liverpool University. Today, I'm presenting our work, Weakly Supervised Learning of RNA Modifications from Low-Resolution Epitranscriptome Data. We study RNA modification. Why? Despite being known since the 1970s, eukaryotic RNA modifications were mostly identified on transfer RNA and ribosomal RNA until the last decade, when they have been identified and characterized on mRNA and various non-coding RNAs. Each of these modifications has its own function, including RNA structure, export, stability, and mRNA splicing. Increasing evidence also suggests that the dysregulation of RNA epigenetic pathways being involved in the pathogenesis of human disease, including cancer. Therefore, it is important to study RNA modification by accurately identifying modifiable sites and methylated genes. Recently, computational approaches have become increasingly popular as a useful alternative to wet lab experiments, especially for preliminary studies. To date, many computational approaches for predicting RNA modifications have been developed, most of which were based on strong supervision, enabled by single nucleotide resolution data. However, high-resolution data may not be available. For example, modifications like HM5C and AC4C were detected by enrichment-based sequencing approaches, where mRNA is fragmented before the immunoprecipitation with corresponding antibody. The immunoprecipitated and import control fragments are then sequenced for reconstructing modifications. Such protocols allow us to identify the RNA modification containing regions of around fragment length resolution, usually about 100 nucleotides. These regions are also known as peaks, enriched with signals of RNA modification. Since a large amount of low-resolution data has been accumulated, our goal here is to identify modifications computationally from low-resolution data. To address the challenges of learning from low-resolution epitranscriptome data, we consider here a weakly supervised learning framework. Weakly supervised learning is aimed at constructing predictive models by learning from data with limited or imprecise labels. We treat the task of identifying RNA modifications from low-resolution data as a multiple-instance learning task, a form of weakly supervised learning where only coarse-grained labels are given. Multiple-instance learning deals with training instances arranged in sets, called bags. A label is provided for the entire bag rather than a single instance level. This formulation is gaining interest because it naturally fits various problems in genomic studies. For example, multiple-instance learning has been intensively applied to study protein-DNA interaction when precise binding site positions are not available. Corresponding to the low-resolution epitranscriptome data, this means that we know only the label of the entire peak region, but it is not exactly clear which part of the sequence covers the modifiable site. A significant proportion of the region may not even contribute to the task. In view of the successful application of deep neural networks in genomic sequence research, we chose to build our model in the way of deep learning. A simplified graphic illustration of the proposed weak IM is given on the right. We first divide the sequence of the entire peak, our bag, into multiple instances. The peak length formed by one single modifiable site can be up to about 2 times the fragment length. When multiple peaks are adjacent, only one long peak is detected, which causes the input size to vary from one fragment length to multiple fragment lengths. Previous RNA modification prediction work had demonstrated that the flanking region of 40 to 50 nucleotides is suitable for identifying modification. Therefore, inherited from the study of protein-DNA interaction, We also use a starting window of length 50 to move along the sequence with stride 10, and divide the sequence into multiple overlapping subsequences, treated as instances. Those biological subsequences are then encoded into numerical matrix and fed into neural networks for further training. Let's take a sequence of 20 nucleotides as an example. A starting window of length 8 starts from the left and extracts a first instance of 8 lengths. Stride 6 means that the start of the second instance is 6 nucleotides after the start of the first instance, that is, the 7th nucleotide. All 3 instances are then represented using one-hot encoding. For example, nucleotide A will be converted to 1000. Convolutional layers are used to extract and score local patches of the input data. The first layer convolutional filters tend to learn relevant sequence motifs, much like a population weight matrix, but without requiring coefficients to be probabilities or log odds ratios. It is worth noting that in the multiple instance learning framework, convolution layer is applied to each instance separately, meaning that the filters start over the subsequence inside of the entire back sequence. The filter weights across instances are shared, so a network searches for the same pattern in all instances. The max pooling layer downsamples the input by taking the maximum value over an input window, which helps to elevate observation noise and identify the presence of longer motif. After the second convolution layer refines the learned features, the hidden representation of each instance is flattened into a feature vector to prepare for the following scoring function. As referred to earlier, in the case of multiple instance learning problem, only an overall label associated with the input bag can be assessed. Therefore, how to obtain bag-level probabilities from instance-level features becomes the key. Generally, we can divide existing score functions into 2 categories: the instance score merging approach and the instance feature merging approach. The instance score merging approach requires the instance-level classifier to estimate the score of each instance. A chosen pooling method, like maximum and average, aggregates all instance scores and returns the bag-level score. However, max pooling only concerns the most favored instance, but may overlook other valuable information. Average pooling assigns equal weights to all instances, which ignores the fact that modifications are sparsely distributed. Although there are other methods, like noise and offers a learnable threshold for averaging, we here consider the second approach, the instance feature merging approach, which aims at obtaining bag-level feature representations using weighted sum along instance representations. We rely on the attention mechanism to learn weight for each instance. The used gated attention consists of 3 fully connected layers. The first 2 layers learns hidden representation using tangent and sigmoid nonlinearity. The third layer learns the similarity between a context vector and the element-wise multiplication between the 2 learned hidden representations. It returns an attention weight for each instance, and the weights are scaled by softmax functions, which ensures that all weights add up to 1. Therefore, the score functions can be invariant to the size of the bag. In addition, the learnable weights allow us to export the contribution of each instance to the bag-level prediction. When the bag-level features are ready, a fully connected layer with sigmoid active functions is used to obtain the final bag-level score and trained using cross-entropy loss. We first validated our model on internal M7G modification. Over 6,000 positive peaks were extracted from both HeLa and HEPG2 cell lines. The negative regions extracted from the area from the same transcript of the positive peak. In order to exclude false positive, only the peaks detected in the 2 cell lines were used. The size and width are balanced between positives and negatives. WICN and its updated version, WICN LSTM, are two multiple instance learning frameworks developed on transcript binding site prediction task. Both are based on instance score merging approach. We reproduce the models on the same dataset as our baselines. The result of a tenfold cross-validation is displayed as the mean value and standard deviation of 7 evaluation matrices. From the table below, we can see that the proposed weak arm framework achieved promising performance and has improved compared to the instance score merging framework. With the help of random cropping data augmentation, the prediction power has been further boosted. One of the advantages of attention mechanism is that it allows to explore the contribution of each instance to the prediction using learned weights. Ideally, the instance that covers a modification should have a specific pattern and contributing most to the back-level prediction. For low-resolution data, since their base resolution is not yet available, we design 5' and 3' margin datasets by extracting two marginal areas connecting the negative and positive regions. Specifically, 600 nucleotides of sequence were extracted for both 5' and 3' side of the identified peaks, with 300 nucleotides within the peak and 300 nucleotides outside the peak. Modification sites are expected to be on the positive half. When single nucleotide resolution data is available, for example for M7G data, we fix the known sites in the middle, extract the flanking region as bags, and visualize the distribution of the instance positions with the highest weights. In that case, the instance containing the intermediate nucleotide is expected to have a higher attention weight. For M7G data, we visualize the distribution of the instance position with the highest weights from both margin datasets. Since the fragment length of M7G mirror-seq is about 80 nucleotides, the density map shows a clear and promising trend. That is, the highest weighted instance of the 3' margin dataset is concentrated in the left half of 80 nucleotides, and the highest weight instance of the 5' margin dataset is concentrated in the right half. For validation on non-M7G sites, we also observed a clear summit in the middle, which indicates the network do tend to assign high weight to the instance cover the true sites. Together, we show the potential of using attention mechanism to detect ion modification at higher resolution from low-resolution data. Interpretability of predicted models is often highly desired for biological systems. In the case of ion modification prediction, this refers to finding the recurring sequence patterns preferred by the model, and elucidating the difference between the high-weight and low-weight instances. We apply the integrated gradients method to quantify the attribution score of each input feature. Since we use one-hot encoding, those attribution scores can be easily converted to nucleotide level. Using several matrices as a reference, we visualize the attribution map of both high-weight and low-weight instances. And it's clear that our model prefers GA-enriched area in M7G prediction task, which is consistent with the existing knowledge of the M7G motifs. To further aggregate instance-level attribution maps and generate high-quality consensus motifs for the target, we apply TF-MODISCO to our results, which extract important k-mers, perform clustering and alignment to generate global sequence motif. Testing on M7G mirror-seq data, we found that the motif discovered by weak arm can match the motif detected by Homer very well, indicating that our framework learns from biological signals and can capture sequence patterns from modifications. We also conducted case studies on HM5C and AC4C. Those two modifications only have low-resolution data. Based on the area under RC curve and the average precession, our model consistently outperforms the baseline models. The validation on 5' and 3' margin datasets also shows the expected results. In a short conclusion, we propose WeakIM, the first weakly supervised learning framework for predicting RNA modifications from low-resolution epitranscriptome datasets. By using the attention mechanism, our model outperforms the previous models in the prediction of transcription factor binding sites. The visualization of the location of instance with the highest attention weight shows the potential of identifying modifications at higher resolutions. And by applying the latest neural network interpretation methods, we showed that our model can capture sequence motifs for target modification. Thank you for listening.